Welcome back everyone. In our last session, we pre-process our microarray data and we perform quality control, data normalization, and prepare an expression matrix. Now, in this session, we will move on to the next step of downstream analysis, that is differential gene expression analysis. Before we start, let me give you a quick overview of the main R packages we will use in this session. First, clear memory to free up space before any analysis. You already know how to install packages from CRAN and Bioconductor. So in this session, we will need few Bioconductor and grant packages. From Bioconductor, we will use AnnotationDbi and HGU133 plus 2.db packages. These are essential for mapping prob IDs to a gene sample that we will discuss shortly. Then we have a Limma package. This is for differential gene expression analysis. We will also use one of its function letter to calculate the average expression values for genes. Next, we need few CRAN packages for data manipulation and data visualization. The dplyr package is very handy for data manipulation tasks such as filtering, selecting, and summarizing data. Along with that, we will use a table package. For visualization, we will use ggplot2 and pheatmap. Now, load all these packages. Now, let's start by looking at the process data we saved earlier. Give me a second to load these dataset. This is our process data. At this stage, we already have the normalized expression data. As you can see, the expression matrix contains prob IDs as raw names and the sample IDs as columns. We know each prob or microarray correspond to a short DNA sequence designed to target a specific transcript or genes. However, probes alone don't provide much biological information because they are simply array specified identifiers, and we need to map these probes to gene samples to interpret our data biologically. If you only have a few prob IDs, you can check them manually using Ensemble Genome Browser. Ensemble regularly update prob mappings for platforms like Affymetrix, Agilent, or Element. You can simply go to Ensemble, type in a prob ID, and see which gene is currently mapped to, including their transcript details and location. Similarly, tools like David also provide prob to gene ID conversion. But if you have hundreds or thousands of probes, like we have more than 41,000 probes in our dataset, so for this dataset, manual searching is impractical. One common source for this prob to gene mapping is the feature data that comes with the array platform. Let's look at the feature data we extracted earlier for this dataset. And this is what feature data looks like. It lists all the probes along with the annotation that was available at the time the array was designed. You will usually see columns like prob IDs, gene sample entries ID, source, and some other information related to that annotation. In our case, the most relevant columns are gene sample entries ID or these gene titles. We can use this information to map probes to a gene. Sometimes we use gene sample for mapping probe to genes, and sometimes we use entries ID and some other gene level identifiers such as ensemble IDs, etc. However, we need to be very vigilant while using feature data because feature data become outdated if the data is from old study or if the annotation file hasn't updated recently. That's why we rely on Bioconductor annotation databases instead. Basically, probes on a microarray chip are fixed. Once the chip is made, these probe sequence never change because these are the physical identifier. However, genome assemblies such that chromosomal sequences and gene annotation information about which probe correspond to which gene can change over time as new functional discoveries are made based on scientific evidence. So, what does that mean for us? It means that a probe that once mapped to a gene one may now correspond to another or may no longer align to any non-gene. Sometimes gene symbols are updated to address some technical issues. A classic example is Microsoft Excel's auto-formatting problem, where certain gene symbols were automatically converted into dates. So, these updates reflect refined genome assemblies, revised transcript structures, and improve understanding of a gene function. That's why using current or updated annotation is important. So this is the key limitation. If we rely only on the old feature data, we risk losing updated information. And that exactly why instead of depending on feature data, we can use annotation database in R. These are provided through the AnnotationDbi package. Basically, AnnotationDbi is a framework or interface that allows us to query annotation database. Each microarray platform has its own chip annotation package. For example, the Affymetrix human genome U133 plus 20 array uses HGU 133 plus 2.db annotation package, a database. Beside platform specific annotation package, there are also organism specific annotation packages. For example, we have this org.hs.eg for human. If you're not sure which annotation package your dataset needs, you can check the annotation slot of your expression set. So check annotation slot. We can use annotation function. In this annotation function, pass the raw data file, pass your raw data file. So this will tell you the annotation name. You can also get the annotation slot if you only run this raw data file. So this will tell you the annotation name. Usually it starts with HGU for Affymetrix. So you can get annotation and can install in the same way we install any Bioconductor packages like this. But here, when installing, remember the package name ends with .db. So for this dataset, we need HGU133 plus 2.db annotation package specific to Affymetrix human genome. Let's check what's inside in this package. We have bunch of information here, so we can retrieve different information from this database. We use this package with AnnotationDbi. Just look at the help page of AnnotationDbi. Basically AnnotationDbi works as a common framework. It provides functions like select to query multiple columns at once, keys to see which identifiers are available in the database, and columns to check what type of information we can retrieve. Also, we have mapID function, mapIDs function to perform one-to-one mapping between probes and gene identifiers. Today we will use the mapID function, which is usually cleaner for downstream analysis. Let's first look at the available columns for our chip. We have lots of columns to choose from. So we use column function from AnnotationDbi package, and let's query our database. We have lots of columns to choose from, probe IDs, the gene samples, uniport IDs, gene name, and so on. Let's check which can be used as keys. So we use key types function from the same AnnotationDbi package. It's look like all columns can be used as keys. We have probe IDs, gene samples, enzyme entries IDs, and other information. So one of the key type is probe ID. That looks right for us. The probe IDs in rows of our process data that going to be our keys that we want to convert into our gene samples. So first, let's extract probe IDs from our process data. These IDs will act as a lookup keys for annotation mapping. Let's break it down step by step. Now we have probe IDs that we map to gene samples. We use mapIDs function from AnnotationDbi package. So the first thing we need to do is specify the database for our platform. Each microarray platform has its own annotation package. So if you are working with different platform, you need to load the respective annotation package here. Next, we need to define the keys. In this case, keys are our probe IDs. These are the identifiers that link the expression data to gene information. So keys equals to probe IDs. Then we specify the key types. Since our keys are probe IDs, we set the key types as probe IDs. If these are gene samples, so the key type will be sample. Now we tell the function which information we want back. Now for us, the column of interest is the sample, that is the gene sample. So column equals to a sample. And the last and most important argument of this mapIDs function is multival. This argument control how we deal with the multiple mapping. So multival equals to first. It is common in microarray data to have multiple probes mapping to the same gene. Sometimes these are actual duplicates added for more coverage, or other times they represent different oligotype sequences from different regions or isoform. So when we map two probes to gene sample, it is very likely that one probe ID will match more than one gene sample. To handle this, we use the multivalence argument. By specifying it, we enforce a one-to-one mapping for each probe. That's way we get only one gene sample per probe row. There are different options. For example, you can choose first mapping that appears, you can filter them, or you can get the list, or you can write your own custom function to decide which mapping to keep. So you have different options to choose. It returns output as character vector. Let's run these lines of code. Now we have a character vector for all the gene samples. To convert it into a data frame, we use deployer function within this code. We are using this pipe operator from deployer package. It is like it sends the output of one command directly into the next command. So in this way, this pipe function make our workflow easier to read. So here we perform three main operations. First we want to convert gene samples into a data frame. Then after converting into a data frame, we want raw names as a column, probe ID. So for this we use table function raw to column to convert raws as the first column. Next we want to rename our gene sample column as samples. So if you look at this gene map df data frame, here we have two columns. One is probe ID and second is gene samples. So by using this pipe operator, you can combine multiple functions together and perform your analysis step by step. At beginning, if this feels overwhelmed, you can do these steps separately. That's totally fine. It's just a formatting thing. Now we have a data where each probe linked to a gene sample. We use these gene samples for differential gene expression analysis. This is the key difference from select function. All the arguments remain same, but there is no this multi-val option for select function. So select function can do similar job, but it returns all possible mappings that often create more rows than your data set, which become messy to manage, especially when you are dealing with large probe set. Let me show you how it works. Everything else will remain same. Let's say samples, and we will use select function from AnnotationDbi. Be careful, we have select function from dplyr package as well, so that's why we are specifying the package name to avoid overlapping of functions. So everything will remain same except multi-val function. So notice that in select function, we have argument columns. In map ID, we have column. We can extract only one column. But if we work with select function, we can extract multiple columns at once. For example, you can fetch gene samples, entries IG, and gene name. Now you can see the output has more rows than your original data set. We have originally 41,000 probes, more than 41,000. But with select function, we get 43,000 rows. That's because some probes map to more than one gene. But for a small data set, you might still get a one-to-one mapping. But for a bigger ones, it quickly becomes hard to manage. And it's turned out to be more complex when you query multiple columns at once. So to keep things cleaner and more consistent when dealing with a large probe set, it's usually better to stick with map ID function. Now you see with map ID, each probe is matched with a single symbol or NA. If there is no match, so some will return as NA. We will remove these rows since they don't provide any information. And you can also see there are multiple probes for a single gene. As I said earlier, this is common in microarray data. This happens for a reason, like some genes are targeted by more than one probe on the array, and some probes might overlap with different isoforms or region of the same gene. Now the question is, how should we handle multiple probes mapping to the same gene? There is no universal answer, and dealing with duplicate is totally a user's choice. People use different strategies for that. Let's go through some common approaches. The first option is to keep the probe with highest expression levels. The idea is that the most strongly expressed probe is likely to represent the dominant transcript or the most active form of the gene. Similarly, people also keep the probes with the high variance across sample. So this is another method which focuses on variance, measure how much a probe's expression change among samples. The same idea we use to remove low expressed probes from our data. So the consideration is that higher the variance tend to carry more biological information, especially when differentiating between different conditions. So variance helps us to avoid keeping probes that are stably expressed across the samples, but the uninformative one. And next we have another method where we take the average of the all probes or multiple probes. So some workflows take the mean or median expression of the all probes mapping to the same gene. This approach assumes that all probes for a gene are reliable and represent the same biological signals. It is simple and often works well when the probe performance is consistent and low variance probes. So we can take the average of the all probes instead of removing all these probes. And sometimes people do choose to remove or duplicate the probes entirely to avoid any potential noise. However, this is more strict strategy and may lead to a loss of useful information, so it should be applied carefully. Now that we understand different strategies for handling multiple probes mapping to the same gene, let's see what our own dataset looks like. Before deciding which approach to use, it's always a good idea to check how many probes map to the same gene. This helps us see whether duplicates are a small issue or a major one. Let's quickly check how many multiple probes we have. So here, first we group our data by gene samples that we have from this gene map data. Then we summarize the probes. This summarize function counts how many probes are assigned to each gene. And finally, we arrange the results in descending order. So the gene with the most probes appear first. If we look at this duplicate summary, you can see we have a cases. We have almost 5,888 probes not mapped to any gene sample. We remove all these probes. And also, you can see we have a cases where up to 14 probes mapped to a single gene. And similarly, we have 12 probes mapped to this gene. And we have a long list of multiple probes that map to a single gene. So let's think about what that means. If we decided to remove all duplicated probes, we would lose a large portion of our probes. Let's check how many total count we have, multiple probes. So this filter function extract all the gene samples that map to more than one probes. If we calculate the sum of these duplicated probes, sum of duplicated genes, so in total we have 32,973 probes that mapped multiple times. If we remove the NA values from this number, that is 32,978 minus 5,888, NA values. So if we decide to remove all these duplicated props that are 27,000, we would lose a large portion of our dataset. That's a huge loss of information at the initial step. So on the other hand, taking only one prop with the highest expression or the variance may not fully represent the variability across all props. As you can see, we have multiple props mapped to a single gene. If we keep only one prop per gene, we lost again lots of data. Like we lost 13 props for this one gene, and similarly goes on for the entire list. So at this point, the best compromise is to take the average expression value of all the props that belong to same gene. This method preserves all the information for multiple props while giving us a single expression value per gene. Yes, it slightly dilutes the signals, but it is a controlled trade-off. We retain the representation of every gene in our dataset instead of discarding data. So let's go ahead and apply this averaging function using Limma package. Let me add the gene samples to the expression data so that we can replace prob IDs with gene samples. First, verify the prob IDs mapping corresponding to expression data. True means the prob IDs from this gene map data frame is matched to the number of rows of processed data. So now we can merge gene samples from our gene map data frame with the process data. This mutate function add a new column with name sample. Now if we check this prob data df, you can see a sample column added next to the prob IDs and expression values. Now we will extend our pipeline. So some probes do not have any gene samples mapping. We can remove those by using filter function. Now we can calculate the average expression for these genes, but before we calculate the average expression for genes, we need to make sure that our dataset contains only numeric value. The columns, prob IDs and samples are the text column character vectors. So these are the character type of a data, not numbers. Since averaging can only be done on numeric data, so we must remove these columns before applying any averaging function. So we will use select function from the dplyr package to exclude them. Here, this minus sign before each column name tells R to remove these columns from our data. Now, at this step, our dataset will only contain numeric expression values for all props, which is necessary before we compute the average expression per gene. Now, let's go ahead and apply this averaging function using a Limma package. This is the example to demonstrate how a Limma function, how this average ref function work. This is our data, X data. If we check out this example, we have multiple rows. We have 2 times A and C. If we apply the average ref function to this X matrix, so the resulting matrix will be reduced from 6 rows to 3 rows. So the similar function will apply to our data to reduce the multiple rows, multiple gene samples actually. Here the argument ID equals to process data df into sample tells R the function to group props based on their gene sample. Basically we specify this column to calculate the average of our expression values. The output now contains one row per gene instead of multiple prob entries. Now we retain total 17,000 probes from our data that we will use for next step. Next, we convert this average data to a standard data frame and then into a numeric matrix, which is the format most analysis function in Limma package required. We can check the structure using structure data to confirm it's numeric. And if we run is.numeric data, it should be returned true, meaning all values are numeric. This step ensures our dataset is ready for the next step, that is differential expression analysis. Now that we have one expression value per gene, let's move to the next step of a differential gene expression analysis. So basically, a differential gene expression analysis is used to compare the gene expression levels between 2 or more sample groups, such as healthy versus diseased tissues or cells exposed to a different liquid. The objective is to identify genes that show significant changes in expression levels between 2 different biological conditions. This helps in identifying potential biomarkers, understanding disease mechanisms, and evaluating treatment effects. In simple terms, we can say that with differential gene expression analysis, we want to identify which genes are upregulated higher in disease conditions like cancer or downregulated lower in cancer. So the first step is the normalization and the pre-processing of the data that we already performed in the last session. The next step is to select a model most suitable for the data. For microarray analysis, linear model is typically preferred. In R, Limma packages is widely used for that purpose. You already learned about linear models and maths behind it in detail earlier in theory session. So here we will apply them practically using Limma package. In order to identify express gene using linear model, we need to do two things. Generate a model matrix specifying the design and fit the model to the design. In all cases, except for the simple two-class comparison, we must also specify a contrast, which we will get to shortly. So start by defining our groups, cancer and normal. We already created a vector for group in previous class, so we will use them here. The groups we are comparing must define as a factor in R because factor tells R that these values represent categories or labels to the particular sample, and the statistical module then know which category is the baseline reference and which one is the comparison group. So in our data, we have two groups, normal, which is baseline, and cancer is our comparison group. And we have already converted them into a factor. Next, we create a design matrix with these groups. Let's run the model dot matrix and then break it down what it means. So what is this design matrix? It represents the experimental setup that your linear model will use. In simple terms, for each gene we fit a linear model. This is similar to performing a t-test for each gene, but we are doing it through a regression framework. We are comparing normal tissues control samples with the cancer tissues disease. So each gene gets its own small regression model. So if you have a 20,000 genes, R will fit about 20,000 models, one for each gene. Then the model can be written as gene expression equals to beta naught or beta 0 plus beta 1 into group. Here, beta 0 is the intercept representing the average expression in the control group, and beta 1 measures how much the expression changes in the cancer group compared to normal group. And this variable group is known as a dummy variable. For normal samples, group equals to 0, and for cancer samples, a group equals to 1. So when we substitute these values, for normal expression equals to beta naught plus 0 into beta 1, 0 equals to normal. And for cancer, expression will be beta 0 plus 1 into beta 1. So for cancer, we have expression equal to beta 0 plus beta 1. That means beta 1 represents the difference in the mean expression between cancer and the normal. Then the statistical test in the model asks a simple question: is the difference beta 1 significantly different from 0? If yes, the gene is differentially expressed between two groups. For more flexibility in our module, we can remove the intercept in the design matrix by adding 0 in our formula. So 0 plus groups. When we do this, the model calculates the coefficient for each group separately. Now we have a separate column for a normal and cancer group. This approach is especially useful when we have a multiple groups. A design matrix without an intercept gives us more flexibility to make a specific comparison between different conditions. For example, in our current data set, we have only two groups, normal and cancer tissues. So we could proceed with an intercept-based design without this 0. But if we are analyzing samples from multiple cancer stages like stage 1 to stage 4, or different treatment groups or different cancer subtypes, a disease matrix without an intercept would allow us to create multiple comparisons within the same framework. So we could easily compare the differential expression genes between stage 1 and stage 2, or we can check out the expression levels of stage 1 versus stage 4 and so on. So depending on what we want to test. In such cases, we simplify remove the intercept from the formula and replace it with 0. Make sure the column names of design matrix correctly match the group names we defined earlier. Now we have column named as normal and cancer for both of our groups. Now we can fit a linear model to each gene. In this case, our data contains gene expression values and design specifies how samples are grouped normal versus cancer. What will happen here? The LM function fit a linear model for every gene, each row in the expression matrix, and it uses the design matrix we created earlier to know which samples belong to normal and which belongs to cancer. Next, here the contrast define a specific comparison you want to test. A contrast is basically a linear combination of the coefficient of our model. In make contrast, this cancer versus normal equals to cancer minus normal defines a contrast where we are comparing sample against normal sample. So cancer is the estimated mean expression for a cancer sample. Normal is the estimated mean expression for normal sample. So the equation, if you recall the equation, that is beta 0 plus beta 1. So we want the expression value of the cancer. So that's why we subtract beta 0 from our beta 1. If we simplify this equation, this comparison, so it is basically beta 1 minus beta 0. So the cancer minus normal compute the difference in expression between cancer and normal for each gene. So here remember one thing, when we use design matrix with an intercept, we don't need to define contrast manually because the baseline group is automatically set and the comparison are made relative to that group. When we create a matrix without an intercept, in our case we replace intercept with 0. So we define contrast explicitly using make contrast function to specify which group we want to compare. So in this way, you can create multiple comparison. For example, if you have several groups such as stage 1, stage 2 and stage 4, you can set a contrast like stage 1 versus stage 2 or stage 1 versus stage 4 and so on using this make contrast function. After creating this contrast matrix, we fit the contrast into linear model. Once model is fitted, we apply the empirical Bayes moderation using E-Bayes function. This step improves the stability of variance estimates, especially when a sample size is small, that is common in a biological dataset. So the E-Bayes function borrows information across all genes to estimate a more reliable variance. It produces moderated t-statistic, which means they are more stable and less affected by noisy or small dataset. After fitting and moderating the module, we can extract the top differentially expressed genes using top table function. When you call it, first we need to specify our module. Then this coefficient equals to cancer versus normal selects the contrast beta 1 that comparing cancer versus normal. Similarly, if you have multiple comparison, you can specify your coefficient as well. We can specify it by numbers as well, like comparison 1. In our case, it's a comparison 1. Instead, this we can use 1 as well. When you have multiple comparison, you can add the numbers as well. Then the number equals to INF infinity means return all genes, not just top genes. You can specify any number as well, like top 100 or top 50 genes. And this adjust dot method equals to BH applies Benjamin-Hodgburg FTR correction to control for multiple testing. As a result, a top table returns a data frame with the columns like log fold change, beta 1 estimates, average expression levels, T means T moderated T statistics, p-value, raw p-values from this moderated t-test. Then we have adjusted p-value, that adjusted p-value from, this is FTR corrected p-adjusted value that we calculate using BH method. Next we have B statistic, that is the log odds for a differential expression. Okay, before we interpreting our results, let's understand what are these p-value and p-adjusted values. First, let's understand what this log fold change value means. Think of fold change as a ratio of expression between two groups. For example, cancer versus normal. You can calculate it like this. A fold change equals to average expression in cancer divided by average expression in gene. Now suppose gene A has 2 times higher expression in cancer compared to normal. That means fold change equals to 2 divided by 1 equals to, that will be 2. So we can say the gene is upregulated twofold in cancer. If a gene shows a 3 times higher expression, it's threefold upregulated. Similarly, if gene B has half the expression in cancer compared to the normal, then the fold change will be a 0.5 divided by 1. That will be a 0.5. That means it's downregulated twofold. But notice something, these numbers 2 and 0.5 are not symmetric. It is difficult to make any comparison between them. So to make it easier to compare up and down regulation on the same scale, we take the logarithm base 2 of the fold change. This gives us the log 2 fold change. So for 2, log 2 fold change equals to plus 1, meaning the expression doubled. And for 0.5, log 2 will be minus 1, meaning the expression half. Now, these values are symmetric around 0. +1 and -1 are symmetric. Positive log4 change means the gene is more expressed in cancer, upregulated, and negative log4 change means the gene is more expressed in normal cancer, normal samples, so downregulated in cancer. If we look at our results table, the log4 change column shows exactly that how much and in which direction the expression changed. You can see positive and negative values here. In our linear module, that beta1 value we estimated earlier represent this log4 change. It tells us the average difference in expression between cancer and normal on log2 scale. And remember, the threshold for log4 change can be adjusted depending on your dataset and biological question. Sometimes researchers use log4 change greater than 1, which mean at least two-fold difference. Other might use 0.5 or 1.5. It depends on how strict you want your selection to be. Remember one thing, log4 change tells us how much the expression of a gene changes between two groups and in which direction. So it tells the magnitude and direction of the expression changes. But for each gene, we also want to know is the difference in expression. The log4 change value is real, or it could be just a random variation in the sample. Like we want to know the significance of the difference. The p-value from p-statistics tells us how likely it is to see a log4 change, log FC this large just by chance, if in reality there was no true difference between cancer and normal cell. So if gene has a log FC equals to +1 but p-value equals to 0.8, that means this two-fold change could easily be random, not a reliable signal. But if gene has a log FC equals to +1 and p-value equals to 0.001, that means this two-fold increase is very unlikely to occur by chance. It's probably a real difference. So in simple terms, the p-value doesn't measure how big the change is. It measures how confident we are that the observed change, the log FC, is real and not a random noise. So in this way, we can get the significance results. We can check the significance of our results. But here is the problem. In microarray experiment, we are not testing one or two genes. We are testing thousands of genes at once. So if we use simple p-value cutoff 0.05, that means 5% of the genes could look significant just by chance, even if there is no real difference in their log FC values. So out of 20,000 genes, about thousands false positive would appear significant purely by random variation. So to fix this, we use something called multiple testing correction. The most common method is the Benjamin-Hochberg procedure, which gives us the adjusted p-value, that also known as false discovery rate. So the p-adjusted value estimates how the significant genes might be false positive. If you use p-adjusted value less than 0.05, it means among the genes you call significant, only about 5% are expected to be false discoveries. So the adjusted p-value gives us more reliable way to select truly significant genes, especially when testing thousands at once. So in simple terms, p-value tells the significance for one gene. P-adjusted value, false discovery rate, is the significance corrected for thousands of genes, that reducing false positive rate. And that's why in practice we filter genes using p-adjusted threshold 0.05 instead of just p-value 0.05 that represent the significance of single gene. And now that we have our differentially expressed results, let's extract the significantly upregulated and downregulated genes from our data. The first step is to apply the cutoff for both statistical and biological significance. So here we are keeping genes with adjusted p-value 0.05 less than 0.05. So here we are keeping genes with adjusted p-value less than 0.05 and log4 gene greater than 1 for upregulated and less than -1 for downregulated. Let's subset upregulated genes and downregulated genes and finally merge them to get all the significant genes. Let's save them into our result folder by using write.csv function. Now we have identified our differentially expressed genes. Let's visualize them. We will start with the Volcano plot. This plot helps us see which genes are significantly upregulated and downregulated. between our two conditions. So the x-axis shows the log fold change and the y-axis shows the minus log 10 for adjusted p-value, which tells us how statistically significant that change is. So here this minus log 10 means that we want to place our significant results, significant gene expression values on the top of the plot. So that's why we use minus log 10 to place our significant results on the top of the plot. And the first line PNG tells R to save upcoming plot as PNG file. So in the similar way, you can save your files as a PDF format as well. And inside this ggplot, we specify our data set that we want to plot. Then we have aesthetics. In this case, the X and Y are the X and Y axis. On X axis, we have log fold change, and on Y axis, we have P adjusted value. And the color scheme, we want the color according to our threshold. These are the significant, downregulated, upregulated, and no, means 3 categories. And this genome point layer creates the actual scatter point. The alpha argument make them slightly transparent, and the size equals to 2 control the point size. So you can adjust these according to your own choice. If you have a large set of a gene, so you can reduce the size. Then the scale color manual defines which colors to use. Red for upregulated, blue for downregulated, and no for not significant, we use gray. You can change these colors as well. And here note one thing, when we are working with ggplot, we use this plus sign to add a layer in our plot. So if I run this line of a command, we created our volcano plot. It clearly highlights which genes show strong fold change and significant p-values. And we have upregulated on the right side with positive values and downregulated on the left side with negative values, and non-significant genes on the bottom of our plot. Once the plot is drawn, this tab off, close the plotting device and save the image. So when you're working with your own dataset, first thing you need to open the device. If I run this line of a command, so it creates an empty PNG file here. And if I run this plot again and use this dev of device of function, so the result will save in this volcano plot. You open the device, you need to close it by using dev of function to save your plot. Now let's create a heat map to visualize expression patterns of the top genes. Heat maps help us to compare expression level across the samples and check the clustering patterns between genes or different groups. For the simple explanation, I am selecting 10 genes at this time. You can adjust the number of genes if you want 50 genes, 25 or 30, whatever you want. Here we will select top differentially expressed genes based on their p-adjusted value, which gives us the most statistically significant genes. Now we extract their expression from the data. So this heat map data is only for the top 10 genes. Now we have to do one more thing. We are interested in checking how our sample clustered together on their expression profiles. So to make the heat map easier to interpret, let's rename the column names. At this time, we have our sample IDs as our column names. So instead of using these sample IDs, we will label them as their actual target groups. So if we look at the phenotype data we have extracted earlier, so the sample 1 is tumor, then we have sample 2 as a normal sample, then again we have sample tumor. So in our data set we have a sample arrangement like this. First sample is from tumor tissue, second is a normal adjacent tissue. Similarly for patient 2, we have tumor tissue, and second we have normal tissue. So we can relabel our sample IDs as cancer 1, cancer 2, normal 1 and normal 2, and so on. If I run these line of commands and check it again, heat data, heat map data. Now instead of sample IDs, we have sample labels. Finally, we can generate heat map. So here the scale equals to none means we already scaled the data manually. We already have scaled data. We perform data normalization, so we don't need to perform any scaling at this step. So scale equals to none. Cluster underscore rows equals to false means we disable raw clustering. We don't want to check the clustering of our genes. We are interested in sample clustering. So that's why cluster columns equals to true. Show names equals to true, display the gene names, and show column names equals to true, display the sample labels. That's why we change our sample labels as cancer 1, cancer 2, and so on to check the clustering. So This heat map gives a compact visual summary of our top 10 genes. You can easily spot which genes are expressed higher in cancer and lower in tumor tissues or vice versa. Now look at the results. Each column is the sample. Each row is a gene. The color represents relative expression levels. Red means upregulated, blue means downregulated, and white means not expressed. And at the top you will see a dendrogram that's showing how sample cluster together. Ideally, the cancer sample should cluster together and normal sample should form a separate group. The dendrogram indicates that most cancer samples cluster together while normal samples form a separate cluster. The large branching distance between the main clusters represent substantial divergence between cancer and normal tissues expression levels. Remember that in our initial QC, sample 8 was indicated as an outlier, but in this heat map, the clustering placed the sample with the cancer cluster. This explains that the single QC matrix is not sufficient to justify sample removal, so exclusion criteria should follow up multiple step verification and validation. So after QC, this is our second step validation that our outlier sample cluster within the same group. So that's all about today's session. In this session, we map probe IDs to updated gene sample using AnnotationDbi package and perform differential gene expression analysis and data visualization. In the next session, we will perform functional enrichment analysis using these differential gene expression genes. Thank you.